Hello Mindsetters, welcome to today's Life Sciences show. Today we have a special revision lesson. We have selected highlights of lessons shown earlier in the term to help you revise Term 1's work. Please post all your questions and comments on our Facebook page on facebook.com/LearnExtra or on Twitter @LearnExtra so we can help you with your revision. Don't forget to download today's show notes on learn.mindset.co.za or click on the link on our Facebook page. Now it's time to get on with our lesson.
Get hold of last year's paper. Get hold of last year's trial paper and a couple of other papers prior to that. Just remember that once you do a section, for example, you do DNA at school, you answer some questions on DNA on your own. So you keep ahead and you keep abreast with what's happening. Study the diagram. Let's going now to the questions. The question 1 says, study the diagram below, which represents a part of a nucleic acid a molecule, and answer the questions that follow. Now, obviously, in today's show, this question will be easier than at the end of the year, because you have only learned one nucleic acid so far. And by the end of the year, or even by next week already, you would have learned the other nucleic acid, and you may have learned other molecules as well. Now it becomes confusing. You've got to choose from those different molecules. But today it's easier. But this is a typical exam type question. So you need to get familiar with this type of work to keep yourself on the toes. Right. So they're giving you a diagram, and they label they're given numbers there, and they give you a key as well. And the key says A is adenine. And obviously G is guanine. First of all, they say identify the nucleic acid shown in the diagram above. And we've just done this work, so this has to be G and A. Remember, you don't have to write out in full deoxyribonucleic acid. DNA is an accepted abbreviation. Okay? Now, I can ask you another question before we even look at the next question here. After you've learned both molecules, another question may ask you, give a reason for your answer. In other words, why are you saying that this is DNA? In this particular diagram, there's only one proof at the moment, and that is that it has two strands. So it's double-stranded. This is not part of this answer, remember. I'm only giving this as an extension to this question. If they ask you, how do you say that this is DNA? Okay? And you've already done RNA, so you understand what I'm saying. Now they're saying label the following: part 1, part 2, and nitrogenous base 3. So part 1 is pointing to this guy here, part 2 to that guy there, and then they ask you for nitrogenous base number 3 there. Okay? Easy stuff. We do it on the diagram. So part 1 is the round one. Remember the outermost part of this structure, the backbone, the side bones there, would be phosphate. Number 2 would be the deoxyribose sugar. I'm just writing the sugar in large so you understand what I'm saying. But remember, whenever there's DNA, you can't just say sugar, you must say deoxyribose sugar. When it's RNA, you must say it's a ribose sugar. Otherwise, you will not get the mark. And nitrogenous base number 3. Now, what is this here? What letter do you see there? The letter A. And this said that this is adenine. Therefore, I said cute girl. So adenine will combine with thymine. So our answer there would be T, or thymine. You write it in full, thymine. What is the collective name for the parts numbered 1, 2, and 3? Let's go back. In other words, this one here, I'm going to use another color there for you. Let's do that. So we're talking about that part there. 1, 2, sorry, not that part. Let's just undo that. Okay. So we're talking about there, 1, 2, and 3. There you go. I said to you earlier, the name for that is called a nucleotide. Nucleotide. Good. Are we together, guys? I'm hoping we're together. Comment on the significance of the nature of the bond labeled for. I love this question. I love to ask this question. Comment on the significance of the nature of the bond labeled for. What do we mean by significance? Significance means, why is it there? What's so good about having this type of bond? So you can't comment on the significance if you don't know the nature of the bond. Let's go back to our diagram. The bond labeled for, not James Bond 007, the bond labeled for. Okay, oops, for some reason the label line is in the wrong place, so it should be labeled 5 in this case here. So we're talking about the bonds there, all these bonds, the bonds between the nitrogenous bases. Okay? So comment on the significance of that. So what do we know about these bonds? What bonds are they? They are weak hydrogen bonds. So what's so good about having weak hydrogen bonds? They break easily to allow for replication. Simple. Look at the question. If it's standing there staring at you, comment on the significance of the nature of the bonds labeled so and so. There's only one bond in DNA molecule that we talk about in really for our study purposes, and that is the weak hydrogen bond. And the word weak is important there. The fact that it's weak, it is easy to break this bond when replication takes place. That means when this double strand separates into 2, and then it can reform again after replication. I'm hoping we're all on the same page there. Okay? The next question says, the diagram below shows part of a DNA molecule in a nucleus just before cell division. Okay? Just before cell division. And they've given you a key there and whatever. But you can see what's happening here. Something's happening, that this molecule is splitting. So identify the parts labeled 3 and 4. Notice the overlap now from what we just did. So in this question, although it's dealing with something else, but it's still dealing with the structure as well. 3 and 4. Let's go to 3 and 4. Okay, there's our 3 there. That would be the deoxyribose sugar. And 4, there is the phosphate. Identify the nitrogenous bases labeled 1 and 2. Whenever they're asking you nitrogenous base, they are testing one simple thing. Do you know that adenine combines with thymine? Do you know that cytosine combines with guanine? That's all they're testing you. So that's what you've got to look for. So number 1, number 2. Let's go to the diagram. Now let's look at the strand. This one was a little bit complicated. If this is the strand and it starts there, then obviously that one combines there, 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 etc., and there. Okay? That's how the molecule was originally. So what was on the other side of number 1 and number 2? Let's look for number 1. Number 1 is this one here. This one here is number 1, there. So where did it come from? It came from that, or was combined originally to that guy. So nitrogenous base number 1 would have to be CG. So it's guanine. Guanine with the G, no? Okay, and number 2, here's number 2. Number 2 was originally, no, look at where number 2 is, right? This is the catch one. Look at where number 2 is. The number 2 is this one here. But number 2 is combining with this guy here. And this guy here was originally combined to C, so it was G. So therefore number 2, sorry, has to be again another, would be cytosine, obviously, because this is G. So this will be cytosine. C. Let's see the logic behind that again. This particular nitrogenous base here was combined to, where am I going? I'm losing my mind here. Let's just undo that before I get carried away there. Okay, so this one here I said was combined with this guy here. Okay, so that was G. So sorry, we're going to go back. And so this will have to be C. Sorry, let me change my answer there. This would be C, not G. So this must be G. Again, another guanine. So we take that out, that becomes guanine. Sorry about that. I looked at the nitrogenous base above. Now, remember, you need to match those bases. And you would have counted 1, 2, 3, 4, and will make it the match that way as well. Okay? Explain why the diagram above represents replication and not transcription. Okay? Why does it represent replication and not transcription? What is happening? What base are you getting here? Thymine. Okay? Thymine. And because of that, this will show that this is a replication and not a transcription, because in transcription involves a messenger RNA, and messenger RNA would have uracil and not thymine. Okay? So, because the nucleotide forming, it has thymine. Therefore, it cannot represent messenger RNA. Okay? Question 3. This is a longer question now. They give you a case study, and they're saying you must answer the question. This guy was investigating the scene of a violent crime. 70-year-old woman. She was stabbed and left to die. They found a few pieces of hair in one of her hands. There was also skin under her nails. They arrested 3 possible suspects. DNA samples were taken from all 3, and hence we come to this point here. So here we're getting the DNA sample from the hair. Here. There's the DNA sample from the victim, suspect 1, 2, and 3. Identify the suspect responsible for the crime, carrying out the crime that is above, in the above evidence there, right? Now, how do we do this? This is a simple matching thing. If we found evidence on the crime scene, and if the suspect has the same DNA, then it's probably him or her. Okay? So what do? We look at this DNA sample. Is it exactly like this one? No. Is it exactly like that one? Already on the top, it's wrong. Is it exactly like that one? No. Is it exactly as that one there? Yes, it does. So this one here is the suspect, suspect 3. And you'll notice whenever they give you a question like this, they always follow it up with provide a reason for your answer above. No matter which case scenario you get, your answer for the second one will always be the same. Why? Because suspect, whichever suspect you choose, because suspect 3 DNA is identical to the DNA of the hair found at the scene. As you're going to grow into life science, you're going to realize that these type of questions are asked and the answer is always the same. Suggest a reason for the comparing the victim's DNA with the other samples. Oh, we got a hair, we got the suspects. Why must we take the victims? We take the victim's hair because we want to compare the sample with the victim, so that we know that the hair doesn't belong to the victim. Otherwise, we look for a criminal till we die. We won't find him because we would have buried the victim. And the last one, do you think that DNA evidence on its own is enough to convict a suspect? No, obviously not, because the hair could have been planted there on the scene. Somebody could have framed this person. There could be mistakes in the lab, and this person could have an identical twin. With that,
all right. Alright, Mindsetters, I hope you guys are enjoying this revision session. Remember, if you are struggling and need help, please post your question on our Facebook page or send an email to helpdesk@learnextra.co.za. Now it's time to take a quick break, so don't go anywhere. We'll be right back. Welcome back, guys. I hope you are getting into the idea of revision. Please let us know how you are doing. I'd love to chat to you on the page or on Twitter. Enough of the chat. Let's get back to the revision.
Guys, obviously we've gone through the summary of what we need to know in this section. Now we need to see how these things will appear in an exam paper, so we can then ask ourselves, are we on track? In other words, this session, and also at school when they give you homework, unless the teacher is just telling you to rewrite the textbook, in which case you must politely tell the teacher that this is not the way to do homework. The homework should be in the form of questions that will test what you have done. Where there are certain cases where you have to summarize other things, that's a different story. But mainly, our focus, especially at grade 12 level, is to sharpen your skills to write the exams, because that is your end product at the end of this year. You need to answer the question. And in order to get a good symbol, you need to understand your work first. So first is the description, the lesson itself, and we're teaching it, and then you're asking the question. So you should be sharp when it comes to questions. And in your possession, you should at least have 3 to 4 past papers. Remember, in life sciences, the papers have been switched around. Paper 1 from last year will be paper 2 at the end of this year with environment. Paper 2. And paper 2 that was written last year will now be paper 1 in the caps. So that's all you can, but the content itself hasn't changed, except for human impact. It's been replaced that whole environment section. You're only doing human impact in environment. You're not doing population studies and social organisms and community structure. You're not doing all of that. You're only doing human impact. But all the types of questions you get are going to be in the same line, okay? So you must remember that. So starting with our question now, tabulate the differences between DNA and RNA. So first of all, we draw a table. When it says tabulate, skill, drawing the table. So in this particular case, you'll notice when they ask you tabulate questions, they always give you an odd number these days of marks. The 1 mark is to draw the table. And the question is given to you, DNA and RNA. So all you do is you draw your table and you put DNA and RNA. So if this was a 5-mark question, you already have 1 mark just for drawing this table. Now that's a skill that you need to carry with you to the exam. You don't have to study for the skill. You have to have the skill. It's 1 mark. So, and when we're looking at this, we're talking, in an exam situation, we would say the structural differences. But here the question is saying tabulate the differences between DNA and RNA. We'll start with the structural ones because that's the important ones, and in the end we'll add others, okay? And notice when you're drawing a table as well, you will have that lines drawn there to show that it's a table. And when you're comparing, when you're talking about something here, what you're talking about on the next side must be relevant to what you're talking about here. Even though it may be correct, but it's in a wrong place, you won't get the mark for that particular one. Okay? So let's start with that. The first one we learned is that DNA is a double helix and RNA is a single strand. That's the first difference. Second difference is that DNA has deoxyribose sugar, and RNA has a ribose sugar. Third difference from what we learned, DNA has ATGC, adenine, thymine, guanine, and cytosine, whereas RNA has A, it has G, and it has C, but it also has uracil in the place of thymine. And I just want to do something here just to show you the difference. So let's use pink there. So there's T. So T has been replaced by U in RNA. Mainly found, now we can go for the other differences, right? Mainly found in the nucleus and sometimes in the mitochondria and chloroplast. So now we're talking not structure, but we're talking about location. And here it's found in the nucleus and cytoplasm. We can even say in the ribosome. But remember that the ribosome is found in the cytoplasm. Just remember that. Okay? Yeah, that's about the important ones. I'm not going to go into other small, niggly differences between DNA and RNA. These are the chief differences between DNA and RNA that you need to know. Sometimes you must also understand there is a document out there called the exam guideline that has been distributed last week to the schools. Please ask your teachers to provide you with that document. That document tells you this is the work that we are doing. This is what is examinable from the work that we're doing. So you need to focus on that before you look at extra. It's always nice to look at extra, but you must first focus on what is important, and then you can do the extra things, okay? Okay, so that's our answer there. Notice what I said earlier, double helix, single strand. Now, if I wrote here double helix and here I wrote ribose sugar, although ribose sugar is correct, but it is not linked to double helix. So you'll get this mark, and this one here will be marked wrong. So remember, you must keep the comparisons that are relevant to each other next to each other. And you must draw the table, obviously. Okay, good. That's the first one done. The other way that they will ask you DNA and RNA and protein synthesis is obviously the diagram below represents protein synthesis. Study the diagram and answer the questions that follow. So they're giving you a diagram showing you the whole of protein synthesis. And again, what I told you last week about a diagram is easier to learn from than from chunks of text. If you remember a diagram like this, or you draw your own diagram to show DNA, to show RNA, to show replication, to show protein synthesis, then that diagram is the one you're going to think about when you're writing. And simply from that diagram, you're going to then carry over your work into your notebook or in your test for that matter. Okay, so keep that in mind. So here they're giving you a diagram. They're showing you that this is the nucleus because the clue there is nuclear membrane, obviously. And the outside the nucleus would be the cytoplasm. This you should know from grade 10. They're showing you there's something there. What is this? The shape tells you what it is. That would be DNA. And they're telling you a process is taking place here, which has resulted in a single strand structure. Okay? They're also showing you these guys here, and they're telling you what those guys are called, tRNA or transfer RNA. And they're showing you this organelle here, and they're labeling it as C, and then they tell you there's the outcome of this whole process. It's a protein that has formed. So I've just gone through the diagram just to analyze. You need to obviously look at the diagram because it's a data that is provided with the question. But you'll do it much quicker, but you're going to scan through it. And make notes on the question if you have to. Let's see what they're asking now. Name the following processes, A and B. A is taking place where? In the nucleus. And that would be transcription. And B is this process here happening here with this transfer RNAs, and that has to be then translation. Describe how the messenger RNA is made from the DNA template during process A. That's already a giveaway to the first question, isn't it? Messenger RNA is associated with transcription, not just messenger RNA, but is made. Messenger RNA is also in translation, but it's not made in translation. It is providing the code during translation. So that's why it's so important, guys, to read the question, because sometimes the clues are there. You just need to focus. Remember, in exams you are nervous, etc. First and foremost, you've got to relax, but you need to know your work. You can't relax if you know nothing. You need to go there prepared. Proper planning prevents poor performance. Okay, how mRNA is made from DNA? So that we have to describe transcription. When does it happen? DNA replication. And I'm going to summarize it. DNA replication takes place, right? While that is happening, you don't have to describe the whole of DNA replication. You say while DNA replication is taking place, messenger RNA nucleotides. Remember from last week, a nucleotide is phosphate, sugar, nitrogenous base. It could be phosphate, sugar, adenine, phosphate, sugar, guanine, phosphate, sugar, cytosine, and in RNA's case, phosphate, sugar, uracil, or in DNA, phosphate, sugar, thymine. These are nucleotides. I mean, these guys are parking there in the nucleoplasm. And while the DNA unwound and separated, unzipped, now it has exposed nitrogenous bases. So what happens there is when this exposure takes place, A, C, G, T, A, when that exposure takes place, instead of DNA molecules coming here, RNA molecules come quickly into this area and they sort of steal the code. So here A is supposed to combine with T in DNA, but there's no T in RNA, so that will become a U. C with G, G with C. T, can we now make that a U? No, T wasn't supposed to be on the other side. T combines with A normally, and there is A adenine in RNA. So you don't make that a U. The U must come where the T was supposed to be, okay? And A would become U again there. So we'll have this messenger RNA moving out there. So that's what's happening there. The messenger RNA nucleotides combine with DNA to form, I would rather say RNA nucleotides, to form messenger RNA. This messenger RNA then separates from the DNA molecule and leaves the nucleus and goes to the cytoplasm and becomes attached to the ribosomes, exposing the codons. The codons are the base triplets. Good. Write down, I didn't write everything there, right? Write down the numbers 1 to 3, and next to each number, the nitrogenous bases that will complete the table. I'm going to do it on the table for you. We're not going to write down. In the exams, obviously you're not going to rewrite this whole table. You will write 1, 2, 3, and you will write your story there. Remember that. You don't have to redraw the table. The instruction told you what to do. So here, number 1, look at what they're telling you. That this is A. This is the first one. On the DNA, it's CAA. So what will it be on the messenger RNA? They want to know. So for C, you will have, let me put it in yellow, you will have G, and A, you'll have U, U. So G, U, U. And what you'll find on the other side, on the transfer RNA, remember that the messenger RNA is opposite to DNA and transfer RNA. I'll explain something just now as well. So if that's G, that becomes CAA. Notice in this case that the DNA and the transfer RNA are the same, because both of them are opposite to messenger RNA, anticodon. Okay? And here they gave you the messenger RNA. You need to work backwards. If that's G, that will be C. If that's C, that will be G. And if that's A, that should be U. And notice again, CGU, CGU. The only time that will be different, the only time that these two will be different from each other is if in this DNA you have an ATT, for example. So ATT will become UAA. And in this amino acid, that will become A, still the same, but now it doesn't have T. So it will have U and U. In that case, only for the difference of thymine, because that's a difference between DNA and RNA, there's no thymine in RNA. So that's the only time it will be difference. So think very carefully. The DNA and the transfer RNA are generally the same, because they are both opposite to messenger RNA. It's like the patty in the middle of a burger. The two roles are the same. The patty is different. Don't write that in an exam, please. You won't get any marks for that. The questions below are based on protein synthesis. Describe the role of DNA during protein synthesis. We've done that already. Or rather, DNA. All right, describe the role. What is the role? DNA provides the code for the formation of messenger RNA. How? During replication, a strand acts as a template. What's a template? It provides the mold for another strand to form, and that's how it works. Notice the important words, the code, template, and during replication this is happening. The diagram below shows the sequence of nitrogenous bases of a small part of a strand of DNA which codes for the part of a protein molecule. So here's sequence of nitrogen of small parts of DNA, not RNA, but DNA. So they're giving you the code there, CGG, TAT, CCT. If they didn't tell you DNA, and they tell you this is of a molecule, why must I tell you now that it's obvious that that's DNA? Why? Because it has T. RNA does not have T. Remember that, that's something that you need to know. Okay, but let's take our big scribbles out there. All right. So we have that situation there. Write down the messenger RNA codon sequence that reads from left to right. Now in this case here, you put your number down, watch it, 3.2. You're reading from left to right, and always keep them in threes the way they are given there. So this will be G, C, C. That would be A, U, A. Remember, the T doesn't become an A, because the T is going to combine with A, and A, there is in RNA. Okay? And the last one, CCT, will become G, G, A. And with that, Luli.
I'm sure you've seen the walk together photo on our page. Please post a caption and like the captions that you think are best. The 3 captions with the most likes will each win a pair of tummy tackies. We'll post the winners on Friday, so get to liking and get all your friends to like too. Now it's time to take a break, so don't go away. We'll be right back. Welcome back from the break. I know you can't wait for the term to end and for the holidays to start. This year we bring you revision sessions from term 1 from the 28th of March till the 6th of April. These will be repeats of all the Learn Extra live shows from term 1. These programs will run from 9 a.m. in the morning till 7 p.m. at night. Go to our website learn.mindset.co.za for all the details. But for now, let's get back to the revision.
Now let's look at what happens during meiosis. And we're going to go through the phases very quickly. This is showing you prophase. Watch what's happening. The chromosomes, just watch as it happens. Remember, in the interphase first, the chromosomes are in single strands. Watch what happens. What happens? DNA replication takes place, and the chromosomes are now formed fully. And that's what we call prophase. And the highlight of prophase is that the chromosomes become visible, and more importantly, the homologous chromosomes lying next to each other. Remember, in mitosis, all these things happen. The nuclear membrane disintegrates, the nucleolus disintegrates, the spindle fiber forms, and the chromosomes become visible. Mitosis and meiosis. But in meiosis, the homologous chromosomes lie next to each other. And as we proceed to the next phase, which is metaphase 1, the homologous chromosomes lie in the middle of the cell, at the equator. Okay? But notice that homologous chromosomes are lying next to each other. Don't be confused with the color. Look at the size of the chromosomes here. The longer chromosomes are lying next to each other, and the shorter ones are lying next to each other. So this one here on the left is a homologue of the one on the right. In other words, one of these chromosomes you got from your mother, and one you got from your father. Okay? During anaphase 1, what will happen is the chromosomes move to opposite poles. Notice we're not saying the chromatids, we're not saying that the centromere broke, nothing like that. We simply say chromosomes. The whole chromosome moved to opposite poles. And during telophase, these chromosomes will be at the poles, and the nuclear membrane will reform, and immediately we go into prophase 2. There you see the chromosomes in prophase 2. And as we move on, you'll notice that the chromosomes start lining up at the equator. Now look, in this particular cell, and I'm glad that they're showing it this way. The other one, we had the middle from left to right. In this cell, they're showing you middle from top to bottom. In a cell, it doesn't matter whether it's side to side or top to bottom. The middle is considered to be the equator. It all depends how the spindles form. So here you have your chromosomes in the middle here. But how are they now? Are the homologous chromosomes lying next to each other? No. The homologous chromosomes are lying independently now. For a start, they've been separated. So each chromosome is lying independently at the equator. Very similar to mitosis. Okay? And what will happen then? The centromeres will break. And watch what happens as we play on. There we go. So where we have anaphase 2, what has happened now? The centromere has split, the spindles have contracted and starting to pull these chromatids apart. Remember, I emphasized in anaphase 1, chromosomes move apart. In anaphase 2, chromatids move apart. Okay? And moving towards telophase, the chromatids are arranged at the equator and as you at the poles, and as you can see, we have now 4 cells with half the chromosome number as the original cell. Original cell had 4 chromosomes. Each of these cells have 2 chromosomes. Let's start with question 1. Study the diagram and answer the questions that follow. Very simple. Instruction there, nothing elaborate about the instruction, but they're giving you a diagram here and they're showing you something happening there. And A, B. Okay. So the first question says, what type of cell division is shown here? What type of cell division is shown here? Okay, in today's time, it's easy because we're doing meiosis. It's easy to answer that question as meiosis. But remember, there's also mitosis that can be tested in an exam. So we are saying what type of cell division taking place there? It's obvious, it's meiosis. Now, obvious as well is the minute they ask you what is this thing here, the next question they're going to ask you is provide observable reasons for your answer. And notice the observable, although it's spelled incorrectly, but it should be observable with an R there, observable reason for your answer. Okay? Although it's spelled incorrectly, but it is in uppercase, normally bold even in your exam paper sometimes. Why? Because they're trying to get to your attention that, listen, guy, we know you know lots of other reasons why this could be a meiosis. We're not interested in those reasons. We are only interested in what you can see in this diagram. And in this diagram, what can we see? One, we can see that the homologous chromosomes are lying next to each other, 1. And 2, we are also seeing this important process here called crossing over. All right, so there we go. Homologous chromosomes are lying next to each other and crossing over is taking place. Any one of the 2 is an acceptable reason. Now they want to know, rest, you're telling us it's meiosis. Very good, you're very clever. Now tell us which phase is it? What is the name of the phase here? And obviously, because of what we said here above, it has to be, according to what we discussed earlier, it has to be prophase 1, because only during prophase 1 do the homologous chromosomes pair up like that and crossing over takes place. Now let's explain what crossing over means very quickly. Crossing over means that the chromosomes that are lying next to each other, this chromosome, you got one from the mother, one from the father, maternal origin, paternal origin, the homologous chromosomes. Each of these two chromosomes code for exactly the same thing. If the one is coding for eye color, size of nose, size of bum, size of finger, whatever else that it could be coding for, the other one must code for the same thing, because you got this set of characteristics from your mother and this set of characteristics from your father. So that's what happens. But when they cross over, what happens? Parts of the one are switched over to parts of the other one. So when this crossing over is taking place, these chromosomes are not as they were before. And this brings about variety in species. That's why you do not look exactly as your brother, AB, or your sister, unless you are twins. Identical twins. This is the variety that we're talking about. Otherwise, we would have all looked like Adam and Eve. Imagine this scenario, AB. Adam and Eve and all their children were all the girls were looking like Eve and all the sons were looking like Adam. And in today's time, imagine that was still the same. I would come to you, AB, hi Adam, how are you? And you would say back,
hi Adam, I'm good.
Okay, and Adam, how is your wife Eve? And what would you say?
How was your wife too, Eve?
There we go. And imagine I'm the cop and you the criminal. And I say, hey, Mr. Adam, can I get your fingerprints? What can you tell me?
No, Mr. Adam, you can't get my fingerprints.
Why?
Because you're Adam.
Exactly, and my fingerprints would be?
Like yours.
So there you go. You can't catch any criminals. So imagine the world without all this. Now imagine that all this was put into Adam and Eve before, AB, this laptop was invented. Before Google. Before Google. In today's generation, if you ask them what was life before Google, they will say, what can we do without Google? Eh? You want to write a speech? Google. You want to find a mate? Google. Eh? The only thing they can't do, we'll talk about next week. Or they can't do on Google. But anyway, even that today you can.
You want to find a Meiosis video?
Google it. There we go. Right. Okay. So imagine the beauty of creation, people. These are moments, while we laughing about it, these are moments that make us think about, that has to be a creator that put all this, has to be some higher power to put all these things without Google. Huh? Okay, what importance process is shown at B? We've discussed that in detail already, crossing over. And what is the significance of the above mentioned process? I told you that already, and that is, it allows for the exchange. Listen to this question and listen to this answer, and I want you to register it. Allows for the exchange of genetic material. And this brings about variety or variation in species. In other words, it makes us different. It makes plants different from each other. All species that are not exactly the same. And this is important because variety means that if there was an ailment and if this ailment had to be in all the offspring, then all the offspring would have that ailment and they would be weak. When we get variety coming in, this now increases the chance of survival within species. Remember, it doesn't mean that you cannot get an ailment because of the variety. It just means that you stand a better chance. If the two partners that resulted in you being born did not both have this characteristic that brings about this ailment. Okay? Let's move on. Draw a fully labeled diagram to show the phase occurring immediately after this phase. So let's go next to this one here, then we know what we're talking about. For a start, what are we talking about? We are talking about metaphase 1. So obviously we will still have the cell membrane. Remember in the exams, when they draw a membrane on the out, it's not the nuclear membrane because the nuclear membrane disintegrates during prophase 1. Okay? So this will be the, let's label it while we're there. Cell membrane. Huh? Okay? Then we'll take these chromosomes. I'm not going to draw them exactly like they are there. I'm going to draw my own chromosomes. And just to show a little bit difference. So what we are showing here is that these are the cell membranes, and then we can put a curly bracket there and say homologous chromosomes together. And if you draw in this diagram, put this like this, together at the equator. So when you draw in this diagram, you will know exactly what this phase is. And guys, the best way of learning meiosis or mitosis or any process is by drawing the diagrams yourself. Draw your own diagrams and draw annotated diagram. Annotated diagram means draw a diagram with notes, meaning if you're talking about it, say a sentence or two, what is happening there. And if you draw this on a double page, take your book, open a double page, and draw the phases of meiosis 1 and the phases of meiosis, or you can do it on a separate double, the meiosis 2, and each one label the diagram. Now don't label chromosome every time, because you've labeled it once, you know what is a chromosome. As you're going on, you're going to label what you see new. More importantly is what you see happening more, or new, sorry. And this series of pictures must tell you a story. So when you're remembering it in an exam, you're remembering pictures, not whole chunks of text, and you explain the pictures you see in your mind's eye. So that's the because this is a diagram we're drawing, we must have a caption. And here, diagram showing metaphase 1 of meiosis. That's what we have to do there. Got it? Great. Okay, so that will be the diagram you're drawing. Good. Let's move to our next question. They're saying here, bacteria reproduce asexually by binary fission. Important note there, not sexually but asexually. And this is by ordinary mitotic division. 2 sterile petri dishes are filled with neutron agar, etc. You got this in your note. I'm not going to read the whole question. All right? Provide a hypothesis. Now remember, although the bacteria is not part of the syllabus in this particular section, however, knowing how to make a hypothesis is. And once you learn the skill of a hypothesis, you'll be able to do that. So provide a hypothesis for this experiment. And that would be, number 1, either ultraviolet light causes mutation or does not cause mutations in bacteria. You don't write both, obviously, in your answer. You're going to write one, because this is a hypothesis. You could have hypothesized that it does, or you could have hypothesized that it does not. You'll have one of those two. Or you could say that bacteria mutate or do not mutate in darkness. Okay? Because of the variables that were there. The important thing is you must have the independent variable. In this case, what can you change? You can change the light. So that's the independent variable. The dependent variable is a mutation, whether the mutation is going to take place or not. And the relationship is this word causes or does not cause. You cannot put the hypothesis in the form of a question. Will ultraviolet cause mutation? That's an aim. That's why you're doing an experiment. That's not what. You must take the chance. You must say it will or it will not. So you must show a relationship. And Amy, the best example we've done before, last year or the year before even, we spoke about a hypothesis. You must have one hypothesis in your head, which you use as a template, and you fit in the new information in there. The one I like to use, Amy, is, if I give her a diamond ring, she will marry me. Let's write that down.
I'm going to steal that one.
Let's write that down. If I give her a diamond ring, I'm not writing it full, she will marry me. Now, Abby, which part are you in control here? Her marrying you or giving the diamond ring? Which part you in control of?
Giving of the ring.
Giving of the ring. So giving the ring is what you, the investigator, can do. So that becomes the independent variable. It can stand on its own. You can either give the ring or you don't give the ring. But whether she marries you or not, It's not up to you. It's still up to her. But it, according to what you're saying, it is dependent on you giving her the
ring.
diamond ring. So this is the dependent variable. And not the whole thing, but marrying the person. Right? Will marry, the will is the relationship. You must take a stand. You can't say, will she marry me if I give her a diamond ring? Because that's a question. You can't say she married me because I gave her, well, that could still be a hypothesis, but in most cases, the past tense will become the conclusion because it's after you've done the experiment. So can you see what we're doing here, guys? I hope you realize we've learned a few skills here. What is the independent variable? What is the dependent variable? What is the relationship? And what is the hypothesis? So I want you to practice that. Don't practice giving the ring. It's not good because she may clap you and run away with your diamond ring. Maybe it's no good. Diamond rings don't come cheap, eh?
Be careful.
Unless you're buying it from those illegal miners there in the other mine there near Benoni. Name the independent and dependent variables in this experiment. And in this experiment, the independent variable, as I said earlier, will be the ultraviolet light because you, the investigator, is in control of that. The dependent variable is the mutation or the change in the bacteria because that you're not in control of. Which apparatus, A or B, was the control and which was the experiment? A was the experiment and B was the control. Let's see why. One petri dish, A, was placed under an ultraviolet light and the other one was left in the dark. We wanted to know whether ultraviolet rays causes this. So the one that we treated with ultraviolet rays, that becomes the experiment, and the one was just left in the dark. That means it wasn't getting this ultraviolet rays. So the one where we remove that factor what we're testing for. What are we testing for? Whether ultraviolet rays causes a mutation. So where we do not use this ultraviolet ray, that becomes the control. Because what if there was a mutation anyway in the ones that were in the dark? That means it was not the ultraviolet rays that caused the mutation. Do you understand that? The logic behind it? Good. What was the result for the control? All the bacterial colonies were red, and that you're getting from the text, guys. You don't have to be a rocket scientist to know that. It's in the text. Explain any two changes to the method that would improve the reliability. Here's another skill we have to learn today. Good, I'm glad we've added another skill here, reliability. Reliability and validity. Reliability means if we repeat this experiment, will we get the same result? Now, let's take that to real life. AB, if I'm a guy that lies to you every time you ask me, something, I lie to you every time. And James, on the other hand, every time you ask him for something, he gives you the truth. Who's reliable?
It's
for
James.
you.
James.
James, you're going to consider reliable. Why?
Yes.
Because every time he's giving the
Keeps
right
his promises.
answer. And with me, I'm giving you all bullshit all the time. Huh? Okay. So therefore, in the same thing, if the result is the same, then the experiment is reliable. It's like marks. Let's say we give a test. I give you a test, AB, and I give you the test, I mark it, I give you 25 out of 50. John marks it, he gets 26 out of 50. James marks it, he gets 24 out of 50. Is the test fairly reliable?
No.
Why? Because it's a 1 mark variation.
Yes.
It's more or less reliable. But if, go the other way, James got 10 out of 50, John got 40 out of 50, and I got 20. Now it's way out, isn't it? 24, 25, all right, there's a slight variation. It's not too bad, but it's reliable. Here now, there's no reliability there, right? Okay? Are we understanding that? Good. So the first thing you must do to improve reliability, you must repeat the experiment. Why? If you repeat the experiment, you should get the same result. Secondly, the other way of doing it is to increase the sample size, because the more sample you're using, you're getting more results, and therefore, repeatability of the result is evident in that as well. Let's move on. The table below shows the percentage relative risk of a mutation occurring during interphase of meiosis at different ages. So they're giving you a table, and you have this in your notes. In age, they're giving you the age, and on this side, the relative chance of a mutation occurring during interphase. And they give you different dates, less than 20, 21 to 40, and so on here, and they're giving you that. important thing here, we need to use the data in the table to draw a histogram. And again, this is also in your notes, so I'm just going to go straight to it. Every graph, remember the 4 graphs that you need to be able to draw: line graph, bar graph, histogram, and a pie chart. So today we're looking at a histogram. The thing about a histogram is ideal for groups of information like this, and notice there are no spaces between the bars. That makes it a histogram. Heading, you must have a caption for all your graphs, everyone, including the independent and dependent variable, to get 1 mark. On this side, you must be able to have interval which is equal, and you must have a heading for this with the unit. If you had that and that together, you get another mark. At the bottom, you must have the intervals. Your bars must be of equal width, and if they were bar graph, they would have to be equally spaced also, but here there's no space. But they must all be equal width, okay? And you must have the x-axis name, which is age, in years. Notice the units come here in this part, not in a heading. Then you would have got 1 mark for all of that. And then for plotting, again, plotting is the easy part. You plot your information and try and get into the habit of writing the plotting so the marker can see that you're doing the right thing. Guys, you cannot just write here 7,5, 18, 37,5, 57, and 74,5. Not allowed. You must have a reasonable scale, and these things must fit into those scales.
We've come to the end of our show. Thank you so much, Mindsetters, for joining me and for chatting to me during the show. Remember to visit our website, learn.mindset.co.za, to get your notes and to watch the videos from the term. Goodbye.
A molecule and answer the questions that follow. Now, obviously, in today's show, this question will be easier than at the end of the year, because you have only learnt 1 nucleic acid so far. And by the end of the year, or even by next week already, you would have learned the other nucleic acid, and you may have learned other molecules as well. Now it becomes confusing. You've got to choose from those different molecules. But today it's easier. But this is a typical exam type question. So you need to get familiar with this type of work to keep yourself on the toes. Right. So they're giving you a diagram, and they label they're given numbers there, and they give you a key as well. And the key says A is adenine, and obviously G is guanine. First of all, they say identify the nucleic acid shown in the diagram above. And we've just done this work, so this has to be G and A. Remember, you don't have to write out in full deoxyribonucleic, not that part. Let's just undo that. Okay, so we're talking about there, 1, 2, and 3. There you go. I said to you earlier, the name for that is called a nucleotide. Nucleotide. Good. Are we together, guys? I'm hoping we're together. Comment on the significance of the nature of the bond labeled for. I love this question. I love to ask this question. Comment on the significance of the nature of the bond labeled for. What do we mean by significance? Significance means, why is it there? What's so good about having this type of bond? So you can't comment on the significance if you don't know the nature of the bond. Let's go back to our diagram. The bond labeled for, not James Bond 007, the bond labeled for. Okay, oops, for some reason the label line is in the wrong place, so it should be labeled 5 in this case here. So we're talking about the bonds there, all these bonds, the bonds between the.
Hello, Mindsetters. Welcome to today's Life Sciences show. Today we have a special revision lesson. We have selected highlights of lessons shown earlier in the term to help you revise Term 1's work. Please post all your questions and comments on our Facebook page on facebook.com/LearnExtra or on Twitter @LearnExtra so we can help you with your revision. Don't forget to download today's show notes on learn.mindset.co.za or click on the link on our Facebook page. Now it's time to get on with our lesson.
Get hold of last year's paper. Get hold of last year's trial paper and a couple of other papers prior to that. Just remember that once you do a section, for example, you do DNA at school, you answer some questions on DNA on your own. So you keep ahead and you keep abreast with what's happening. Study the diagram. Going now to the questions. The question 1 says, study the diagram below, which represents a part of a nucleic acid. DNA is an accepted abbreviation. Okay? Now, I can ask you another question before we even look at the next question here. After you've learned both molecules, another question may ask you, give a reason for your answer. In other words, why are you saying that this is DNA? In this particular diagram, there's only one proof at the moment, and that is that it has 2 strands. So it's double-stranded. This is not part of this answer, remember. I'm only giving this as an extension to this question. If they ask you, how do you say that this is DNA? Okay? And you've already done RNA, so you understand what I'm saying. Now they're saying label the following: part 1, part 2, and nitrogenous base 3. So part 1 is pointing to this guy here, part 2 to that guy there, and then they ask you for nitrogenous base number 3 there. Okay? Easy stuff. We do it on the diagram. So part 1 is the round one. Remember the outermost part of this structure, the backbone, the side bones there, would be phosphate. Number 2 would be the deoxyribose sugar. I'm just writing the sugar in large so you understand what I'm saying. But remember, whenever there's DNA, you can't just say sugar, you must say deoxyribose sugar. When it's RNA, you must say it's a ribose sugar. Otherwise, you will not get the mark. And nitrogenous base number 3. Now, what is this here? What letter do you see there? The letter A. And this said that this is adenine. Therefore, I said cute girl. So adenine will combine with thymine. So our answer there would be T or thymine. You write it in full, thymine. What is the collective name for the parts numbered 1, 2, and 3? Let's go back. In other words, this one here, I'm going to use another color there for you. Let's do that. So we're talking about that part there.